Okay. So what's a cell? Cell is the fundamental, structural, functional unit of all living things. Do you know what's a living thing? Okay. Living thing could be uh, bacteria. Remember, except virus. Virus is not living. It's non-living. All right. Bacteria, plants, animals, fungi, everything is living. So all living things except virus. Virus is not a living thing. Okay, so learn this definition. What's the definition? A cell is the fundamental, basic, structural, and functional unit of all living things. Okay, Sharon, what's the definition of cell?
A cell is the fundamental,
Mhm.
structural, and functional unit of all living
Yeah,
organisms.
Sneha.
A cell is the fundamental, structural, and functional unit of all living organisms.
Yeah, Advaita.
Sir, a cell is the fundamental, structural, and functional unit of all living organisms.
Organism. Okay. Now, you might have heard the name of these scientists. Anton von Leeuwenhoek. Okay, Anton von Leeuwenhoek. He is a scientist who first observed and described a cell, right? And who discovered the cell? He, Robert Hooke. These names you have to learn, especially for NEET exam. You must memorize all these names. Okay, they are in the reader itself. So for school exam and NEET exam, it's the same stuff, except some extra questions. That's all, right? So keep learning. So who discovered the cell? Robert Hooke. Who observed and described the first living cell? Anton von Leeuwenhoek. Okay? And you know, he took slices of cork. You would have seen cork used in the wine bottles. It's from a tree, right? Some of the you might have seen, champagne. When they open champagne, they just flip the cork. This cork is from a tree. So he took slices of this cork and looked at the microscope developed by Leeuwenhoek. Okay, and he saw tiny spaces. These spaces are known as cells. Sometimes for the prison they use the term cell. Okay, cell means a tiny space. These tiny spaces are known as cells. Okay? So who discovered the cell? Who discovered the cell? Robert Hooke. Who observed and described a living cell? Anton von Leeuwenhoek. So how do you observe the cell? We have two kinds of microscope. The first one is light microscope, and the second one is electron microscope. Okay, and in light microscope, we use white light. Okay, from a lamp or sunlight. In electron microscope, we use electron beams. We use electron beams. Okay, what's the advantage of using electron beams? Electron beams are very short wavelength. So these microscopes have very high resolution. Okay, that means you get very clear images and it has very high magnification. This is the advantage of electron microscope, right? With electron microscope, you can see every part of the cell, okay? And with light microscope, the one in your school, there's a light microscope. It's white light and it has low resolution. What's the meaning of resolution? So we don't study it now, but understand it. Resolution, like you might have seen, HD, high density pictures, and low quality pictures. High density picture has high resolution, clarity, and the other one doesn't have. So here you get more detailed image, and here magnification is less, low magnification, low magnification. So how do we see the cells? Today we see the cells with light microscope. This light microscope is also called compound microscope. And what's other microscope? The electron microscope. Okay, we don't study about this, but you have to know that there are two kinds of microscope. Okay, after the discovery of cell by Robert Hooke, two scientists. These are the names of the scientists. They are German scientists. Okay, it's called Schleiden. Schleiden. It's called Schoen. Schleiden and Schoen. And they created a theory, right? Now this name, Schleiden and Schoen. These are two scientists. What do they propose? They propose that all living things are made up of cells. Okay? All living things are made up of cells, and cells arise from an existing cell. This is called cell theory. They propose cell theory. It was a new discovery those days. Today we know living things are made up of cells. It's not a big deal. But those days it was a very big discovery, right? Okay. What Scleden did, he observed that plants are made up of cells. Okay. And the Theodore Schoen reported that there is something called cell membrane, plasma membrane, okay, in plant cells. And they both proposed the cell theory. So let's look at cell theory. What does cell theory? What does cell theory says? Okay. And that's another scientist, Rudolf Virchow. Rudolf Virchow explained that cells divide and form new cells. Okay. One cell divide and form new cells. Okay. So, you study in class 9, 10 that cells undergo mitosis. But think about 1855. Those days, no one knew about these things. Okay. So he is the one who found out that one cell divide and form new cell. That means a pre-existing cell forms a new cell. Okay. So he called it omnis cellula a cellula. Okay, it's Latin. What does that mean? That means new cells are formed from the pre-existing cell. Okay, and with this idea, the cell theory was, modified. Okay, what was original cell theory? They said that all plants and animals are made up of cells. This is their cell theory. Okay, but after the discovery of Rudolf Virchow, it was modified. Now, what's the new cell theory? All organisms are made up of cells and the products of cells. And all cells arise from pre-existing cells. All living things like humans, we are made up of cells or products of the cell. Like we have products of the cell, like bone. Bone is made up of cell and cell products. Blood is made up of blood cells and plasma. So we also have products of the cell, and cells arise from the pre-existing cell. Okay, learn this one. These are two statements of cell theory. Okay, and learn the name of the scientist, Rudolf Virchow. What he did, he explained this. This is in the reader, so you have to learn this. Okay, omnis cellula is cellula. What is it? Omnis is cellula, right? Omnis cellula is cellula. Okay, and what's the name of these two scientists? Schleiden and Schoen. Right? Today, cell theory is known in their name, and this is a complete cell theory. The two statements of cell theory. All living things are made up of what? Cells and the products of the cells. And all cells are formed from pre-existing cell. Okay, there's no magic like all of a sudden a living thing appears. No, for a living thing to form, there should be another living thing. There should be a parent. A parent cell only forms a daughter cell. Okay, there's no magic here, right? For a cell to be formed, there should be a previous cell. This is the cell theory. Okay, can you say the cell theory, Advaita? Can you hear me?
Um, all cells arise
Yeah,
from pre-existing cells,
yeah. All living things are, yeah. All cells arise from pre-existing cells. What's the other one? All living things are
and
made
all
up of
living organisms are made
cells
up
and
of
products.
cells and products of
Cells. Okay. Yeah. Learn one more time. All living organisms are made up of cells and the products of cell, and all cells arise from pre-existing cells. Okay.
Yes, sir.
Yeah, Sharon.
All living organisms are composed of cells and the
Yes.
product of cells.
Yes. Okay.
And all the cells are
All
formed,
living things. All living things.
all living things are
They're composed of cells and products. What's the
cells
other
and
one?
products of cells.
All. Yeah. Can you
All
repeat?
cells arise from the pre-existing cells.
Sneha, try.
All living organisms are composed of
Mhm.
cells and products of cells.
Yes.
All the cells exist from pre-existing cells.
Cells. Okay. Yeah. Now this is like overview of cell, right? We'll be studying in detail about it. If it's a plant cell, in plant cell, you studied there is a cell wall and there is a cell membrane. Okay, there's a cell membrane and there is cytoplasm inside. And there is a nucleus. Nucleus contain genetic material. That's plant cell. How about animal cell? Animal cells have cell membrane. Animal cells have cytoplasm, and it has a nucleus with genetic material, right? So what difference you find between plant cells and animals? Plant cells have cell wall, but animal cells don't have. Okay, there are many more. We'll study about each part. So this you have to memorize. This is like what are the three things here? Plant cells have cell wall, but plant cells and animal cells have cell membrane, cytoplasm, and nucleus. Okay, so that's the first point. First 2 points. What's the first 2 points? Plant cells have cell wall. Okay, plant cells and animal cells both have cytoplasm, cell membrane, and nucleus. Look, what's the smallest cell? This you have to learn. Smallest cell is mycoplasma. What is mycoplasma? Mycoplasma is a cell, bacterial cell without, see this one is mycoplasma. This is mycoplasma. It's a bacteria without the cell wall. Remember, bacteria has a cell wall. So mycoplasma is the smallest living cell, and it's similar to bacteria but without cell wall. That's an easy way to memorize. And what's the length of this? 0.3 micrometer. Do you know what's a micrometer is? Okay, like 1 centimeter is how many millimeters? 10 millimeters. Okay, so times 10, you get, right? 10 millimeters times 1000, you get 1 micrometer. Okay, 1 micrometer. So you need to know micrometer. What's the next one? In physics, we'll study this nanometer. So this is 10 to the power 6 meters. 10 to the power, 9 meters. So 10 to the power 6 meters and 10 to the power 9 meters. Physics will be studying this, but we need mainly micrometer. Okay, how do you change millimeter to micrometer? Times 1000. If it's centimeter, times 10,000. Okay, so that's 0.3 micro. This means micrometer. Micrometer. Okay, smaller than millimeter, right? And how many micrometers? 0.3. And what's the largest cell? Ostrich. Okay, ostrich egg is almost 10 to, it varies between 10, 15 centimeters. It's huge, right? Egg of ostrich. Remember, one, chicken egg is one cell. When you crack an egg, look at the shell, and below the shell, you can see a thin membrane, plastic-like membrane. That's a cell membrane. And its cytoplasm has a lot of protein stored. And that protein is the white part, and then this yellow part. And if you crack it in a plate, preferably in a black plate, And look at the center of the egg yolk, you find a spot with a different color. That's a nucleus. Okay, so there is egg yolk, and then there is egg white, and there is cell membrane. Okay, that's a cell membrane. And exactly in the center, you can see a nucleus. This nucleus will form a chicken if when it hatches. So what's the largest cell? One cell, one egg is one cell. The largest cell is the egg of ostrich. Remember, what's the longest cell? It's a nerve cell, right? And size of the bacteria, 3 to 5 micrometer. Look at this one. Mycoplasma is zero point 0.3, right? So it's almost 10 times smaller than the bacteria, 0.3. And red blood cells, this you did in class 10. It has biconcave shape. What's the diameter of this? 7 micrometer. 7 micrometer is the diameter of human red blood cell. These are in the reader. We need to learn this, okay? And right. And eukaryotic cell. Look at eukaryotic cell, like it's larger, 10 to 20 micrometer. Viruses are not cell, okay? Well, they're not cell, 0.02 to 0.2 micrometer. And PPLO. PPLO stands for pluro pneumonia like organisms. Okay, you can use PPLO, but that's what it says. It's P, pluro pneumonia like organism. Okay, it's almost same as a mycoplasma, right? And that's like 0.1 micrometer. And typical bacteria is 1 point. Now the problem is this is a figure in the reader, but these are the ones written in the text. Okay, so learn the one in the text rather than like typical bacteria is 1 to 2, but here it is 3 to 5, right? And virus you can learn from here. PPLO is mycoplasma, so learn this one and learn the eukaryotic cell from here. Okay, so it's different. In the diagram they say something, and in the text sometimes they don't match. Okay, so learn the one in the text. What's written in the text? These are different size and shape of cells. Right? Look at the size and shape. Some are like disc-like. These are plant cells. We'll be studying them in other chapters, so no need to learn anything now. And these are different kinds of animal cells, right? It can be disc-like, polygonal, column-like, cube-like, okay, thread-like or irregular. See the nerve cell, muscle cell, bone cell. Okay, these are like cardiac muscle cell, heart muscle cells, sperm and the egg, all the blood cells, white blood cells, red blood cells, glands. Okay, here you find parenchyma in plants, collenchyma, sclerenchyma. Okay, don't worry about the names now. What does it mean? It means the cells come in different shape. Okay? This shape, polygonal, column-like, cube-like, thread-like, okay, irregular. Usually, they don't ask any questions based on this, so you can skip that. So, what are the two kinds of cells? Cells can be divided into two categories. We can divide the cells into two different categories. Let me connect. Okay, these are types of cells. Types of cells. Okay, cells are classified into two categories. What's the first kind? It's called prokaryote. And the second is called eukaryote. So what are the two kinds of cell? Prokaryote and eukaryote. What's a prokaryote? It's a cell where you don't find a nucleus. There's no nucleus. Okay, but here you find this one has nucleus. Here no nucleus, this has nucleus. And this one has membrane bound. Okay, membrane bound organelle. Means what? The parts of the cell are surrounded by membrane. For example, mitochondria. You know this from last year, right? Mitochondria. In around mitochondria, you find a membrane. See, this is a mitochondria. You can find a membrane around it. So it's called membrane bound organelle. But here, no membrane bound organelle. Okay, what's the meaning of bound? Means covered by. When you buy a chocolate, it's bound by a plastic covering, or paper covering. Okay, so no membrane bound organelle. Right? So this is the main difference between the prokaryote and eukaryote. What's the example for prokaryote? Bacteria is one example. Mycoplasma, the one we studied now. Okay, or they also call this PPLO. These are examples of prokaryote. And eukaryotes, what are the examples of eukaryotes? Plant cell, animal cell, okay, and fungi. Okay. Fungi is like plant cell, but slightly different. So every living thing is eukaryote. Remember, virus is not a cell. Virus is non-living and it's not a cell, right? Okay. So these are two kinds of cell. Learn this one. What are the two kinds of cell? Prokaryote and eukaryote. What's a prokaryote? A prokaryote, you don't find a nucleus. There is no membrane bound organelle. But in eukaryote, you find a nucleus. Nucleus is present. It is membrane bound organelle. Like mitochondria. What's the example? Mitochondria. What's the example here? Bacteria and mycoplasma. Okay, or PPLO, right? These are the two types of cell. And virus is not a cell. What's the reason? It's non-living. Virus is non-living, so it's not a cell. Tell me the types of cell, Advaita.
Sir, there are two types, prokaryotic cells and eukaryotic cells.
Yeah. What's the difference?
Eukaryotic cells have no nucleus,
No, the other way.
and
Other way.
sorry.
Prokaryote,
Eukaryotic cells
nucleus.
have a nucleus and
Yes.
have membrane-bound cell
Organelle.
organelles, while prokaryotes do not have a nucleus, and there is no membrane-bound cell organelles.
Yeah.
Examples of eukaryotes are mitochondria and animal cells
Mitochondria
or
is
plant cells.
an organelle, yes. What's the example for eukaryotic cells?
Plant cells, animal cells,
Yeah, plant cells and animals.
fungi.
Yes. What's the example for prokaryote?
Prokaryotes, mycoplasma or PPLO.
Or bacteria, right? Even bacteria. Yeah. Sneha, what's the difference between prokaryote and eukaryote?
Sir, prokaryotes have no nucleus and no bounded cell organelles.
Membrane bound cell
Membrane-bound cell organelles. Eukaryotes have nucleus and membrane-bound cell organelles. Example of prokaryotes is bacteria and mycoplasma.
Yes.
Example for eukaryotes is plant cells, animal cells, fungi.
Karen.
In prokaryotic cells, there is no nucleus and no
Membrane
membrane-bounded
organelle.
organelle.
Mm-hmm.
In eukaryotic cells, there is nucleus and membrane-bounded organelle.
Yeah.
And example of prokaryotic cells is bacteria, mycoplasma, and eukaryotic cells example is fungi,
Yeah.
mitochondria.
Okay. Mitochondria is organelle, right?
Yeah.
Okay. Hello. Can you hear me?
Yes, sir.
Okay. Yes. And let's study the structure of bacteria. Okay. Structure of bacteria. A bacteria is a prokaryote. Okay. And in bacteria, you find that bacterial cell is surrounded by a protective covering. They call this glycocalyx. What is it? Glycocalyx. Okay, glycocalyx is of two types, right? Glycocalyx could be a slime covering or it could be a capsule. Okay, a slime covering or capsule. So, what's the outermost covering of glycocalyx? Covering of bacteria, glycocalyx, and this is for protection. Okay, protection from dryness. It help the bacteria to stick to each other. Okay, this is the outermost covering. What's outermost covering? The glycocalyx. Glycocalyx. Okay, it's mainly for protection. There are many functions. Learn at least one function now and this name. And there are two types. One is loosely attached, and the second one is tightly attached. Okay, the loosely attached is called slime. Other one is called a capsule. Right, and the next layer is a cell wall. Okay, cell wall is made up of a substance known as peptidoglycan. What is it? Peptidoglycan. Peptidoglycan is a chemical. Peptide. Peptide means amino acid. Glycan means glucose. When you learn the names, always try to break it up, simplify it, and memorize. Okay, peptidoglycan, made up of a combination of amino acids and glucose. Right? And what's a cell wall of plants made up of? If you remember, it's made up of cellulose. Cell wall of fungus, it's made up of a substance called chitin or chitin. But cell wall of bacteria is made up of peptidoglycan. Okay, keep saying the name. These are new terms. It takes time for you to memorize a name. So the only way is keep saying, repeat it. New names. Keep saying in your mind, peptidoglycan, peptidoglycan. Okay, peptidoglycan, made up of amino acids and glucose. It's a combination of amino acids and glucose. This is called peptidoglycan. And on the cell wall and cell membrane, you find different structures. Okay, there is something called pilus. Pilus is a structure which help bacteria to perform sexual reproduction. How do bacteria perform sexual reproduction? During sexual reproduction, the two bacteria with the pilus come close to each other, and through this, a copy of plasmid. We'll study now what is a plasmid. A copy of plasmid go to the other side. This is sexual reproduction. Okay? There's no egg and sperm formed here, right? So what's the function of pilus? Sexual reproduction. During sexual reproduction, the genetic material, the copy of plasmid is transferred to the other cell. Okay, this is sexual reproduction. That's pilus. Then you find some other structures known as long filament-like structures. These are called fimbriae. Fimbriae help bacteria to stick to any surface, stick to any cell surface, any cell, plant cell, animal cell, or even on non-living surface. Okay, so what's the function of fimbriae? For sticking to any surface like cells. What's the function of pilus? Sexual reproduction. Okay, so let's start from the outermost covering. Outermost covering is glycocalyx for protection. It can be loose one or tightly attached one. Okay, you can call it slime, or the other one is like a capsule. Number two, what's the second layer? Second layer is cell wall. What is it made up of? Peptidoglycan. What's the function of the cell wall? It gives shape to the cell. No, bacteria comes in different shapes. We'll study it now. And it prevents bursting. Prevents bursting of the cell when the water enters. Cell should not burst. That's done by the cell wall. What is cell wall made up of? Peptidoglycan, which is a compound made up of glucose and amino acids. And this is called pilus. Pilus for sexual reproduction. Okay. And the next filament-like structures are known as fimbriae. What is it? Fimbriae. Fimbriae help bacteria to stick to any other cells. Okay. And this is another structure. I'll show you the detailed picture of this. But right now, let's study what is it. This is flagellum. Hope you know the structure of this one. Flagellum is for movement. A flagellum has a basal granule that controls its movement. Inside the flagellum of bacteria, you find 9 microtubules. So what is made up of? 9 microtubules. The picture I'll show you. Learn this part. Flagellum, and it has basal granule, and it's on the cell membrane. Okay, it's on the cell membrane and the cell wall, connected to both. Right? Okay. This is the cell membrane, and this part is the cell membrane. So what are the three layers from outside? Starting from outside, what's the first layer? Keep memorizing. Okay, outermost layer, glycocalyx. Second layer, cell wall. Third layer, what's the third layer? Cell membrane. This is cell membrane. Okay, this is cell membrane. Three layers. What's glycocalyx? For what is it for? Protection from many things, back from antibiotics, dryness. Okay, so mainly you learn dryness from the dryness. It can be loosely attached or tightly attached. Number two is cell wall, made up of peptidoglycan. It gives shape to the cell and prevents bursting of the cell. Okay. And number three is cell membrane. Okay. There are three layers. But on the cell wall, you find some extra things. Pilus. That's one. What is pilus for? Pilus for sexual reproduction. Okay. You don't need to learn the process, but learn the function. Pilus for sexual reproduction. Okay. Then there is fimbriae. It helps bacteria to stick to any other cell. And the third one is a large structure known as flagellum. Flagellum for movement. Okay. And don't learn the microtubules, learn the function. This is for movement. Okay, that's for movement. And what controls the flagellum? The flagellum is controlled by a basal granule, which is on the cell membrane and the cell wall. They both together hold the flagellum. Flagellum for movement. Okay. Can we review this? Advaita, can you say the outermost layer?
Um, Glycocalyx.
Calyx. Okay. And what's the use of glycocalyx?
It is used for the protection of the cell from dryness.
Yeah. And What are the two types of glycocalyx?
It is slimy and like loosely.
Yeah, one is tightly attached and the other one is loosely attached. Learn only that part, okay?
Yeah.
Yeah. What's the
Yeah.
second, next layer? Cell wall. What's the next
Yeah,
layer?
cell.
Cell wall, yeah. What is cell wall made up of?
Peptidoglycan.
Peptidoglycan, okay. And what's the function of this?
It protects the cell from bursting
Bursting.
when water enters.
Okay. And what are the three structures you might find on the cell wall?
Pilus.
Yes. For
It is used for sexual reproduction.
yeah. And what's the next one?
Fimbriae.
Embryo. For
Like attaching to another bacterial
surface.
cell.
Yeah. And one more structure.
Flagellum.
For movement.
Movement.
Okay. Yes. Aaron. So all the parts we study.
The outermost part is called as the glycolic
Calyx.
calyx,
Glyco.
and it is loosely attached
Attached
and
or
tightly attached.
tightly attached. If it's loosely attached, what
It's
do you call
called
it?
slime.
Slime. Okay, or
If
slime
it's
layer. Oh, yes.
tightly attached, it's called capsule.
Capsule. What's the function of this layer?
So what?
What's the function of glycocalyx?
It prevents dryness.
Dryness means protection from dryness. What's the next layer?
Next layer is cell wall.
Cell wall. And what is cell
And
wall
it's made
made
up of peptidoglycan.
Glycan. And what do you find? What's the function of cell wall?
It prevents from bursting,
Bursting and
and
gives
it gives
shape.
shape to the cell.
Yes, give shape to the cell. And what are three things you find on the cell wall?
Pilus,
Embryo,
fimbriae, and
flagellum. Flagellum.
flagellum.
No, flagellum.
Flagellum. Yeah.
Haven't you studied flagellum in class 10? Like sperm has
No.
a sperm. What's the tail of the sperm? What did you study in class 10? No, you studied human reproduction, right? In class 10? Last year?
Yeah.
There was sperm and what's the other name of the tail of the sperm? Do you remember that? It's called flagellum, right?
Oh.
Yes. Okay. What's use of flagellum? Movement.
Movement. Yeah.
Movement. Okay. Yeah.
It's controlled by basal granule.
Basal granule. Yes. What's the third structure?
Pilus,
Oh yeah, pilus. For
sexual reproduction.
yeah. Did you say about fimbriae?
Yes.
Okay. Yeah. Sneha, name all the parts we studied.
The outermost covering is glycocalyx. It gives protection from dryness. It is loosely attached and tightly attached.
Yes. If it's
Then
loosely attached, what do you call it? Slime layer.
slime, yeah. If
Yeah, and
it's
the
tightly attached, it is capsule.
capsule. Yes. What's the next layer?
Cell wall, peptidoglycan.
Yes.
It gives shape and prevents
Bursting. Prevents bursting, right?
yeah.
What's the next layer?
Pilus used for sexual reproduction.
Reproduction. Yes.
Fimbriae, for like help sticking to another cell.
Yes. And the long
Then
tail
flagellum,
flagellum. Flagellum for what?
movement.
Movement. Good. Okay. Then you find cell membrane, right? Cell membrane, there's a little more details. I'm not going to tell it here, but as we move up to next slides, we'll study about more details. But the cell membrane is folded inwards. Can you see? It's folded inwards, right? So this inward folding is called mesosome. Okay, and there's more features of mesosome. I'll explain in the upcoming slide. But what's the use of mesosome? Mesosome is for cell division. Okay, at least one you learn. There are more. We'll be learning it. Okay, and it helps in respiration. Learn two features. Mesosomes are infoldings, infolding of cell membrane. Okay, infolding of cell membrane is called mesosome, and it help in cell division. Means division of the bacteria plus respiration. Okay, what's a jelly-like substance? It's called cytoplasm. Cytoplasm. In cytoplasm, all chemical reactions happen. And do they have a nucleus? No. Then what do you find here? You find a circular DNA. This is called a circular DNA. Sometimes it's also known as nucleoid. Okay, this is called circular DNA. It's DNA, but just one ring of DNA. There is no nuclear membrane around it. In some bacteria, you find a small ring of DNA, which is called plasmid. Okay, both are made up of DNA. Plasmid contains extra genes. It carries extra genes. Another organelle you find here is known as ribosomes. Okay, what is this? Ribosome. Okay, this ribosome is called 70S ribosome. That's the name of the ribosome. I'll explain this. What is 70S? Okay, it's a small ribosome. It's called 70S ribosome. What's the name of the ribosome? It's called 70S. 70S ribosome. And function of 70S ribosome, protein synthesis. Okay, protein synthesis. And there are more parts called, cell inclusions like glycogen and all those things. But as due to limitation of space, I'm not writing them. Okay, and you will get confused. So let's learn all of them. Only these many things. There are more things coming up, but learn only this, right? Starting from outside. What's the outermost layer? Outermost layer is okay. Uh, outermost layer is glycocalyx. What is it? Glycocalyx. And glycocalyx gives protection. And you can see two types. Some bacteria has, it's called slime layer. Okay, we call it slime layer. Okay, and the second one name is capsule. Slime layer or capsule. Slime layer is loosely stuck on the cell wall. Capsule is tightly stuck on the cell wall. And it's for protection. Protection mainly from dryness. And number two, what's the second layer? Cell wall. What is cell wall made up of? Peptidoglycan. It gives shape to the cell. Different bacteria have different shapes, and it prevents bursting of the cell. And the third layer is known as cell membrane. Number three, the cell membrane. Cell membrane, what's the function of cell membrane? Cell membrane controls movement of molecules in and out of the cell. Is a function of cell membrane. It regulates what goes into the cell, out of the cell. And the cell membrane is folded inside. Infolding of the cell membrane is known as mesosome. Mesosome is the infolding of the cell membrane. And this infolding performs these two functions, cell division and respiration. It performs cell division and respiration. Okay? And there are a few more functions and details of the structure. We'll be doing it. And what is this? Circular DNA. What does DNA do? It controls cell activities. Okay? Cell activities are controlled by the circular DNA. And there's plasmid. Plasmid has extra genes for like antibiotic resistance. Okay? It has extra genes which give bacteria antibiotic resistance. What is pilus for? Pilus is used for sexual reproduction, flagellum for movement, and fimbriae to stick to any other cell, maybe bacterial cell, another bacterial cell. Ribosomes are known as 70th ribosomes. 70th ribosomes for protein synthesis. And there is cytoplasm. What is cytoplasm? In cytoplasm, the chemical reactions happen. Metabolic reactions. It's a jelly-like substance. Metabolic reactions happen in the cytoplasm. Okay, so these are the parts of a typical bacterial cell. Okay. If you have any questions, please ask. All right. Hope you can learn this one. Right. Yeah. Look at this. These are prokaryotic cells. What are the examples for? What are the features? They have no membrane-bound nucleus and organelle. Means no nucleus and no membrane-bound organelle. Examples: bacteria, blue-green algae. Okay, it's all cyanobacteria, mycoplasma, and PPLO. And that's the expansion of PPLO. Pleuro, pneumonia-like organism. Okay, the slight difference between them. We'll do it later. They're like mycoplasma. It's in another chapter. Okay. And they are smaller in size. They reproduce quickly, multiply rapidly than eukaryote. They are different shape and size. Okay, this you have to learn. This already we learned this. Go to the next page. These are the shapes of bacteria. Bacteria can be rod shaped. Okay, if you remember, there was a bacteria there's a bacteria in yogurt. It's called lactobacillus. Bacillus means rod-like. If it's spherical, it's called a coccus. Okay, spherical means coccus. Vibrio means it looks like a comma. So comma shape, right? Right. Vibrio. Spiral, like it's spiral shaped. Spiral shaped, spirillum. Okay. Bacillus, coccus, vibrio, and spirillum. Bacillus, like rod shape, spherical, coccus. Okay, coccus. Vibrio, comma shape. Spirillum, spiral shape. Okay, so here you can see this is what's the shape of this. You see, it's like this is rod shape, right? Bacillus, and that's spherical, round. This is like the comma. Okay, comma shape. This is spiral shape. Yeah. Okay, tell me the shapes of bacteria. Sneha, say the four
Uh,
shapes of bacteria. Yes.
Rod shaped, which
Mm.
is bacillus.
Yeah.
Sphere shaped, which is coccus.
Mhm.
Um, Coma shaped, which is mm,
Vibrio.
Vibrio.
Vibrio. But something similar to this, Vibgyor. Vibgyor is 7 colors, right? So Vibrio.
Yes.
Yeah, Vibrio. Yes. What's the last one?
Then spiral shaped.
Spirillum.
I don't know.
Okay, I'll ask you. What's a comma shaped? Yeah.
Vib,
Name the. What do you call comma shaped?
vib,
Vibrio. Vibrio. Vibrio.
vibrio.
Keep repeating. That's the only way you can, learn these names. Otherwise, you keep forgetting. Keep saying in your mind. Maybe it's 5 times, 6 times. It will stick. Okay? Vibrio. Vibrio. Right? What do you call the spherical ones?
Sterilium.
Spherical. Sphere. Caucus.
Oh yeah, good.
Okay. Yeah. Learn one more time. Learn what's a rod-shaped bacillus. Caucus. Spherical. Put those pictures in your mind. Those, violet spheres, right? Caucus, spherical. And comma shape, vibri. Spiral shape, spirillum. Isn't it? That sounds similar, right? Spirillum, spiral shape, spirillum. Okay, yeah. Advaita,
Yes, sir.
yeah. Name the rod-shaped bacteria.
Bacillus,
And comma shape
Vibrio,
and spiral shape.
Spirillum,
Mhm. And what's the last one?
the sphere shape, that is coccus.
Caucus. Yeah. Taryn, say all the names. Spherical,
Uh,
sphere shaped. Yeah.
Rod-like is bacillus,
Mm-hmm.
spherical is coccus,
Yes.
comma-shaped is vibrio,
Yeah.
and spiral is spiral.
Very good. Okay. Sneha, one more attempt.
Yes, sir. Rod shaped is bacillus.
Yes.
Sphere shaped is coccus.
Mm-hmm.
Comma shaped is vibrio.
Mm-hmm.
And spiral shaped is spiral.
Yes. Okay. So these are the parts of the bacteria. We studied most of them, but we'll be doing with little more detail, right? So we studied cell envelope. What's the name of cell envelope? Glycocalyx. Okay. It can be of two kinds, a slime layer, or it can be capsule, slime layer or capsule. Then we studied about mesosome, which is infolding of the membrane. When the membrane is folded inside, it's known as mesosome. Chromatophores will be studied now. Okay, this is in some bacteria for photosynthesis. Nucleoid is a DNA. Okay, DNA loop is called nucleoid. Flagellum we studied, but more details are coming. Pili means pilus. Pili is plural, pilus singular. Fimbriae we did, ribosomes we did, and we'll be studying inclusion bodies. Okay, these are 7 parts we need to know. Okay, cell envelope is covering of the cell. It's called glycocalyx. Mesosome, infolding of the membrane. Chromatophore for photosynthesis. Nucleoid is a circular DNA. Flagellum for movement. Pilus for sexual reproduction. Fimbria to attach to any surface. Ribosome for protein synthesis. And these are extra things present in the cell. Now look at the next part. That's a cell envelope. Can you see from outside? It's called glycocalyx. This one. Okay. Glycocalyx. Second layer, cell wall. Third layer, plasma membrane. Okay. What is plasma membrane? It's a cell membrane. Cell membrane is known as plasma membrane because it surrounds cytoplasm. So plasma membrane, cell wall, glycocalyx. Okay. And these are 3 layers: glycocalyx, cell wall, plasma membrane. You already learned this one. And this is the pili. Then there's plasmid and ribosomes. Okay. That's glycocalyx. its outermost layer, and it can vary in thickness. But what are two types? Slime layer, loosely attached. Capsule, tightly attached. This is thick and tough, right? So these are the two types of glycocalyx. And cell wall is the next layer. It's in every prokaryote, give shape to the cell and prevent bacteria from bursting, okay, by giving support, right, or collapsing. Plasma membrane. It's what's a property of plasma membrane? It's partially permeable, semi-permeable. Means what? It allows only small molecules to pass through. It allows small molecules to pass through. That's the meaning of semi-permeable. Okay, and it's similar to the eukaryotic cells. Okay, and you can identify the bacteria by putting stain. This, stain. What's a stain? In school, you might have done the onion peel or cheek cells. When you do the cheek cells, you put a blue colored liquid on that. Okay? This is a stain. What's a stain? It's a colored solution. And because the cells don't have any color, it's called stain. So what is stain? Stain is a chemical to give color. Okay. Hope you can hear me.
Yes, sir.
Yeah, okay. And so what's a stain? Stain gives color to the cells. Okay. And this scientist, Christian Graham, he developed a method of staining. It's called Gram staining. We don't learn the steps, but he colored the bacteria with specific stain. And if the stain is present, it's called Gram from his name, Gram positive. If it's absent, he called it Gram negative. Okay. Gram positive gets a darker color, gram negative as a lighter color. Okay, and in gram positive, then takes up the stain and retains most of the stain. Gram negative do not retain the stain. And why do they do this? This is for identifying the bacteria. Like when you go to the hospital, suppose someone has an infection, they check what kind of bacteria is it. It's one way to find the bacteria. Okay, they stain the bacteria and check for identification, and it also helps in classification of the bacteria. So, what's the name of the procedure? Gram staining. What are the two kinds of bacteria according to gram staining? Gram positive, gram negative. What's gram positive? If they retain the stain, if they retain the gram stain, it's called gram positive. If they do not retain the gram stain, it's called gram negative. Okay. Questions if you have. Good. Okay. Yeah. Right. Can you explain gram staining? What is gram staining? Advaita.
So, staining a particular cell to identify it.
Yes. And what are the two types of bacteria according to the staining properties?
Gram positive and gram negative.
Negative. What's the difference between gram positive and negative?
Gram positive retains the stain,
Mhm.
while gram negative does not.
That's not. Okay. Yeah. Sharon,
Yes, sir.
what is gram staining?
Uh, it is a chemical that's used to give the color.
To the bacteria. Okay. If the bacteria retains a stain, what do you call it?
Positive,
Okay. Gram positive. And what's the
gram
other
positive,
one? Gram negative.
gram negative.
Yeah. Sneha.
Yes, sir.
What is gram staining and why do they do this?
It gives color to a particular bacteria to like identify it.
Yes, yes. Okay. Now look at mesosome. Let's some more details. What is mesosome? Infolding of plasma membrane. Infolding, folded inside. Okay, it's called infolding. If you look at the infolding, you can see structures like tube-like structures, round structures, vesicles, and they are in layers. So they call it lamellae. Lamellae means in different layers, right? So what are three parts of mesosome? Vesicles. Vesicles means round structure. Tube-like structures. Tubules. Lamellae means in layers. Okay, lamellae, right? So this is mesosome. What's mesosome? Infolding of plasma membrane. It's infolding of plasma membrane. Okay, and what's the function of mesosome? Number one, cell wall formation. Help in cell wall formation. And replication means division. Help in division of DNA. Okay, and separation. Distribution means separation of chromosomes means DNA to the daughter cells. It helps in respiration, secretion, and increases surface area of cell membrane. When it's folded, it has large surface area. So what are three parts? What is mesosome? Mesosome is infolding of plasma membrane. What is it? Infolding of the folded inside. And what is it made up of? Okay, what are the parts? Parts of the what are the parts of this? Parts of the mesosome. Vesicle, tubule, lamellae. Okay, vesicle is round structures. Tubule is tube-like structures. Lamellae in different layers. Okay, when it's in different layers, it's called tubule means tube-like. Okay, so these are three parts. Then the functions: cell wall formation, division of DNA. It's called replication. If you don't know this, learn this one. Replication of DNA and separation of DNA to daughter cells. How do bacteria divide? Look at this. When the bacteria divide, if you remember, it divides by binary fission. Okay, and this is the mesosome, and the DNA stick to the mesosome. With the help of mesosome, the DNA become two. You have two DNA. Then what happens? The mesosome separate the two DNA to the two sides. Okay, that's what it says, separation of DNA. And then a cell wall is formed. So it's the DNA. And in the middle, a cell wall is formed with the help of the mesosome. So this is cell wall. Okay, and these are the two DNA. Okay, that's what they say. Function, formation of cell wall. When when do they form the cell wall? During binary fission. And replication, division of DNA, separation. You see separation? Separation of DNA to the daughter cells. These are two daughter cells. These are two daughter cells. Okay, and it contains enzymes. Remember, there are enzymes inside. These enzymes help in respiration. Okay, and if bacteria want to secrete, means release anything outside, release any substance outside. It's called secretion, and it's done by the mesosome. And that's is it folded up, so increases surface area for all enzyme reaction. Okay, increases surface area of plasma membrane for enzyme reactions. Okay, enzyme reactions. This is mesosome. What is mesosome? Infolding of plasma membrane. And what are three parts of mesosome? Vesicles, tubules, and lamellae. Lamellae means in layers, right? What does vesicle means? Like, spherical, spheres, bags of membrane. Tube-like, tubule. Lamellae, in layers. Okay? You can see here, mesosome is like in layers. What does it do? Help in cell wall formation, separate the DNA, and help in division, replication of DNA. It has enzymes for respiration, okay, respiration, and it help in secreting things outside and increases surface area. Okay, so there are many functions, and try to say as many as possible. Okay, yeah, ready? What's mesosome, Advaita?
So lysosome is formed in the infolding of the plasma membrane,
Yeah, and why is it?
and it is of
Mhm.
three types.
Yeah,
It has three parts:
yeah,
vesicles, tubules, and lamellae.
yes, and say some functions, as many as you can.
Okay. So it helps in the cell wall formation,
Yes.
and it is used for DNA replication.
Mm-hmm.
It is used in the distribution of the chromosomes to the daughter cells.
Yes.
It is used for respiration and secretion,
Yes.
and it is used to increase the surface area in the plasma membrane and enzymatic content.
Yeah. Okay. Good. Yes, Sneha.
Yes, sir.
So, what is a mesosome?
Formed by the enfolding of plasma membrane.
Hmm. And
It
can you
has parts,
say the parts here?
vesicles, tubules, and lamella.
Lamellae. Lamellae. Okay. Yes. And say some function.
It helps in the formation of cell wall,
Mm-hmm.
in the division of DNA.
Mm-hmm.
It helps in increasing the surface area, uh, sec- uh, respiration and secretion.
Yeah.
And
Yeah. Okay. Good. Yeah. Sneha. Try. What is mesosome?
sir, I said right.
Pardon? Oh, you said.
Yes, sir.
Okay. Yeah. Look at the chromatophores, right? And what's chromatophore? This is like chloroplast. Chromatophores, uh, membrane. It's surrounded by a membrane, and it contains pigments like chlorophyll, and it can absorb the light. And using light, it does photosynthesis. It does photosynthesis. This is chromatophores, right? Remember, there's no chloroplast, but some bacteria. What's the name of bacteria? Cyano. Cyano means blue. It's called blue-green algae or cyano. Cyano means blue, right? Cyanobacteria. Cyanobacteria has again membranous infolding, and they have shown it here. Some of the membrane is infolded, but it has pigments. What do you find inside? The pigments. What the pigments do? They absorb the light, and the light is used in photosynthesis. Okay, I think we'll stop here. Too much information. We'll continue tomorrow, right? Tomorrow, can we keep it at 2 o'clock? Do you have any
Yes.
tuition tomorrow? Because in the morning, I found out that many of you have, maybe you can sleep in the morning, and some of you have church or programs in the morning. Can we keep it at 2, like Advaita? Is it okay for you?
Mm, yeah, sir. I think it's fine.
Are you free at 2 o'clock?
Actually, someone has called me to my friend's house, so I thought after, like in the morning, I'll come for tuition and then go.
Mm.
So I think it'll be fine though. I can come.
Yeah. Can you go in the morning or after the tuition?
Yeah, sure, sir.
That's fine. Yeah. How about Sneha?
Yeah.
Sir, I have a class at 2:30.
Oh, okay. What tuition is that?
It's a dance class.
Oh, dance class. 2:32.
4:30.
Mm-hmm. Okay. Mm. Evenings you go out, right? With the family.
Yes.
Mm. So I'll keep it in the morning. It's okay. 11 o'clock. It's fine. 11 is okay, right?
Yeah.
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 11 or
Yes.
12. What do you like? If you want to sleep, I can make it like 11 or 12.
11 is fine, sir.
Is it okay, 11?
Yes,
Yes,
sir.
sir.
Yeah, okay. So 11 o'clock, right? I'll send you the notes. Try to learn it. I'll send you the presentation notes. Maybe even a recording I can send you. So you can, review it, right? Yeah. Thank you very much. See you
Yeah.
tomorrow.
Okay, sir.
Okay. Bye-bye.
Thank you, sir.
Bye.
Thank you, sir.
Bye. Bye.